Hi everyone, my name is Brianna and my partner's name is Sam. And we are going to present our presentation, the identification of bacteria in soil through biochemical testing. So bacteria are unicellular organisms that are present everywhere around us. For this lab, we use soil, and 1 gram of soil can contain thousands of bacterial species. Through testing soil, we can predict biodiversity, bioactivity, and biomass.
So for our hypothesis, we conducted a series of stains, and we hypothesized that if we did conduct these stains, differentials, and biochemical tests on an unknown organism from a soil sample, then we can thoroughly identify the species of an unknown organism.
So the methodology of our lab was basically gathering our soil sample, which we then performed a serial dilution of. This serial dilution was plated on multiple agar plates, including 10% TSA, 50% TSA, BHA, all culture plates. For this lab, we chose bacteria from the all culture plate, bacteria D as labeled with the purple arrow. And with this bacteria, we performed gram stain analysis, endospore analysis, antibiotic analysis, and biochemical tests.
So for the analysis and the results of the finding, we take our results and we compare it to the results of known strains of bacteria. And by comparing these, we're able to see the similarities and the differences to determine which strain most closely aligns with our unknown isolates. And our results show that through a series of biochemical tests and analysis, we found that the bacteria chosen from our soil sample is Neisseria gonorrhea.
So as Sam stated, we had a table basically showing the differences between our isolate and various known bacteria. From this, we concluded that biochemical tests are useful in the identification of bacteria within a sample. Future work can include replicating the biochemical tests that we've performed to ensure that we conducted them correctly.
So for the results, we start out with the gram stain analysis, which indicated by the circular picture, we can clearly see that it is a gram negative stain or result, excuse me. And for our interpretation, we interpreted that this indicates a thin peptidoglycan cell wall.
Based on the stain, we looked at the morphology, which then we determined that it had a cockeye shape.
We further researched into the endospore stain analysis, and we saw that the stains were pink / a red color, and this indicated a negative result, which further indicated that there were no spores present and that we only had vegetative cells.
We performed sugar fermentation biochemical tests with 4 different sugars. For glucose, we found that it was positive for acid and negative for gas. For lactose mannitol, we found that it was negative for acid and positive for gas. For sucrose, we found that it was negative for acid and negative for gas.
The next test we're going to discuss is the oxidase test, which a positive test result is indicated by this purple color shown in this image down below it, where there is no purple color for the negative square. And our isolate showed a positive oxidase, so it looked like the positive square, indicating there's a presence of cytochrome C.
Our next test was the KOH biochemical test, which we added a drop of KOH to our bacteria. And when we did this, it became very sticky and stain-like, as you can see in the picture. And from this, we determined that it was positive for gram negative.
The next test completed was the nitrogen reduction test, and we saw a clear color change from the clearish yellow type of color, transforming into this bright red with no gas in the Durham tube. This indicates a positive nitrate reduction, but no nitrogen gas formation.
So for our acid tolerance test, we added acid to our bacteria on our agar plate, and our bacteria didn't grow, meaning that it was negative for growth.
And the last test on this slide is the citrate utilization. Our isolate was positive for citrate utilization, meaning the agar slant changed from a green to a blue color, indicating that our isolate can use citrate.
For our starch hydrolysis test, we flooded our agar plate with iodine. And when we did this, our bacteria had a very big clearing, meaning that it was positive for amylase hydrolyzation.
The next test was the gelatin test, and our isolate was positive for gelatinase. So after conducting all of the steps and putting the broths on ice, our isolate still did not harden after the allotted time, meaning that there is gelatinase causing this like fluidity within our broth.
For our blood test, we had a gray yellowish bacteria after letting it sit for a few days, and there was no clearing around, meaning there was no hemolysis caused by H2O2, and we determined that it was alpha hemolysis.
The next test was the phospholipid test using the egg yolk agar plates. And our isolate had a slight clearing around it, which indicates that it's positive for lipase to break down the phospholipids in the egg yolk.
For the EMB biochemical test, we found that it was negative for lactose fermentation.
And lastly on this slide is the MSA plate. Our isolate showed that it was negative for mannitol fermentation, and the MSA plate and the EMB plate were good tests to conduct because our results were consistent with the results obtained in the sugar fermentations test for lactose and mannitol, further proving our results. So onto the interpretation, what we did was we conducted this table by charting our isolate results in the second column here, listing all of the biochemical tests on the far left, and we compared it up against 5 different strains of bacteria. But all of these 5 bacteria are all gram-negative cockeyed bacteria.
From comparing these to our isolate, we found the fewest differences between Neisseria gonorrhea and our isolate, with only 3 differences compared to the rest that had 6 or more. So we concluded that the bacteria that was isolated from the soil collected was Neisseria gonorrhea.
And that concludes our presentation. Thank you, everyone.
Thank you.